Thank you for the introduction, and again, thank you to the organisers for asking me to talk. So as you've heard, I'm a clinical pharmacologist, so a few perhaps perspectives and I have to say personal thoughts on some of the clinical issues related to early development of, well, really the targeted type of nanomedicine. Qualifications for this, I guess, are this was something that I started having to look into last year as the company was getting into the siRNA area, and we had a molecule potentially coming towards the clinic. So I can offer description of some of the challenges. I have to say I'm not sure I can offer many solutions, but perhaps that's something we can work on together. So in terms of the, let's get my own pointer out because I know this one doesn't work. Okay. A few definitions. I thought I'd better get clear on what we're talking about, or at least what I'm going to talk about. So when it comes to the nanomedicine, I mean, basically, I think what I'll be talking about is sort of the delivery system approach. Some of which are very old, well, relatively old technologies, but obviously some of which are much newer, and they can deliver just about anything as far as I can tell. We've been hearing about quite a lot of things in this meeting. I wasn't going to say very much about this sort of any specific nanomolecular type approaches, unless you consider antibodies to be a nanomedicine. Maybe we can come back to that one later. So I'm thinking here of delivery systems, and in the specific case it's siRNA, and the systems necessary to deliver it in a targeted way. And then what do I mean by targeted? I don't need to say, I mean, we have systems, if you like, of the sort of almost the passive targeting to tissues. It could be either of those. And then there's the active approaches of, some sort of molecular targeting system that might deliver the systemically administered agent to the tissue or cell of interest. There are also, we heard about some of this yesterday, local approaches. Again, I'm thinking just about this, systemic administration, possibly topical administration for the gut, but delivering to the tissue of interest. In terms of targeting, are we talking about delivering it to the tissue, or are we talking about increasing the uptake into cells? I guess it could be either of those. And this is one that I think we should come back to in the panel discussion later. Is it targeting a tissue, because that's what I hear most of the discussion about so far, or is it targeting a particular subset of patients? And from the therapeutic point of view, it's mostly targeting a tissue. I guess this is something I think we ought to come back to in the panel, because this actually might be the bigger issue. And from a medicine point of view, that maybe is less, nanomedicines may have less to offer here. It's obviously the diagnostics that could be the opportunity. In terms of the general challenges, we heard yesterday uniformly from regulators around the world, essentially nanomedicines are no different than conventional medicines. They're going to have to deliver the same things, and Bernd was talking about some of this earlier. Yes, there will have to be a demonstration of safety in the clinic, and that will be for the payload, for the carrier, and potentially for the device. And I don't see any reason to think that's going to be different requirements from what we have to do now just for a systemically administered molecule. Likewise, efficacy. Why would the measures of efficacy be any different if you're treating the same disease with a targeted nanomedicine versus a totally untargeted small molecule? The efficacy measures still need to be the same, and they need to be things that, will A, get your drug registered, but B, get the patients and the prescribers actually, believing and that it will do them some good and willing to take it. Quality. Bernd has mentioned this already, particularly for the CM&C side. These requirements are going to be the same. And increasingly, at least in Europe, but I'm guessing in the US as well in the next few years, cost effectiveness, it's going to be the same as for a small medicine. Getting into the more specific challenges, the issue, or the potential opportunity perhaps, is the highly targeted drugs or interventions, targeted particularly to a tissue, organ or cell. Especially because linked in with this, and nanomedicine is sort of beginning to offer us the opportunity to give rather more complex agents with a more complex mechanism of action, perhaps a more indirect mechanism of action, modifying, the siRNAs, gene therapies, and so on. These can be very different from standard, the conventional small molecule or even many biologicals. Essentially, the thinking is, as long as we maintain the drug level at a particular systemic level, there will be drug at the site of action and it will work. And we use systemic exposure as our surrogate, if you like, for efficacy. And we aim to dose so that we maintain an exposure. But is that the right thing to do with some of these more targeted agents? You know, would systemic exposures actually help us? Because the question that I started asking myself was, if we get, how are we going to pick a dose of a targeted medicine, particularly one that's highly targeted, where there may not be any systemic exposure, or very little, and it may be very indirectly associated with efficacy? Why should I care? Well, if you get the dose wrong, particularly if it's too high or too frequent, you may undo all the benefits of the targeting, because you'll begin to get the non-specific effects kicking in again. So I started thinking about that a bit. And as I've said, the conventional PK/PD type approaches may not be all that helpful. So we're going to need to use, rather more in the way of model-based approaches, I think. Here you have an advantage in this field, because there's some very nice work already being done. So this is work from Bartlett and Davis, which I'm sure many in the audience are aware of. But building, looking at what the biological system is, delivering the drug systemically, obviously in this case to a mouse or a rat, passage to site of action, and then uptake, across the cell membrane, avoidance of the endosome, into I can't see the slides properly, but very carefully describing the molecular basis of the mechanism of action and building a complex biological model with a complex mathematical model built on top of it, with, I think it was 29 different parameters involved in that model, and they can do some very nice things with it. So here's the data, here's just some examples from one of their papers, data, and then the model fit, taking this data and understanding and including it in their model and adapting the parameters so that they correctly describe the data. So, a dose response curve, rather nicely, I think, and rather importantly probably for those of us who might actually pick clinical doses, profiling what the effect of the doubling time of the cell is. And the shorter the doubling time, so the faster the cells proliferate, the shorter the duration of action of the drug, of the siRNA. This is siRNA specific. From a dilutional effect, you just get more cells. And then taking that information and being able to start look at the effect of dosing regimens, not just single doses, but what would we have to do in order to maintain the effect at our level? It can get very complicated, particularly in oncology. If the effect of the drug is to increase the doubling time, i.e. decrease the rate of proliferation, you have a very dynamic situation in that the dose regimen you might start off with might not be the dose regimen you end up with. It gets complicated, but if it's the right thing to do, we're going to have to find ways of dealing with it. But this is obviously a complex model where the parameters all have to be worked out from in vitro and ex vivo experiments. So one of my colleagues, Tim Goggin, pulled off the shelf some work that he had done about 5 years previously, describing, this was work to look at the effect of a cytotoxic on hemopoietic cells, so particularly platelets, an indirect drug response. And he dusted that model off and applied it to siRNA. It's a much simpler model, only 5 parameters that you can actually completely identify from the pharmacodynamic data, and he proved this, or demonstrated this at least, by taking data from, this is a Nature paper on ApoB in rodents, I think, and he was able to build a model just taking the data from the paper, and on the right-hand side you've got the model fit. So a much simpler approach, very fewer parameters that you can pull off just from the experimental data. And the reality is probably we're going to need to use both types of approaches to help us in understanding things like doses for these highly targeted drugs. So there may be a way forward, but it's going to be difficult. And then just to finish, last slide, a few more challenges over and above that. And I guess I'm intrinsically an optimist. So if you work in the drug industry, you probably have to be an optimist these days. Otherwise you'd get out and do something else. But I'm sort of working on the basis that we will get some molecules through. So as we begin to get the efficacy and the safety piece sorted out, and we begin to get doses and drugs into the clinic, targeted nanomedicines into the clinic, you have to start thinking about a few more questions. When you look at a drug label, very little of it is about efficacy. Okay? Quite a lot more about in the content of a drug label is about safety, but about half of it is about how you change the dose to deal with the patients who wouldn't actually have fitted in your clinical trials, but do exist in the real world and do need to be treated. Organ failure, drug interactions, etc. So, can we start thinking about how we're going to change the dose in different types of patients? What about drug interactions? It probably won't be the conventional ones, but are there going to be any surprises? You know, might there be both desirable and undesirable drug interactions through mechanisms completely different from the classical mechanisms of drug interaction? I don't know, but I think it's possible. Likewise, drug-disease interactions, particularly in these dynamic states where the disease may be changing and the impact on the drug may change over time. And then lastly, this was sort of hinted at by Bernd, with the fact that changing the formulation, changing the device, we have to, the conventional approach to dealing with that in the clinic is simply, do you get the same systemic drug levels with the new formulation, the new device, the new route of administration, as with the old one? If you do, fine, you can assume the same safety and efficacy. So this is the bioequivalence approach. What we're going to do if and when, because in my world it always is when, not if, the device changes or the formulation changes, how are we going to demonstrate bioequivalence there for some of these more targeted medicines? I don't have an answer. I hope in a few years' time we'll be at the stage where it's actually really important to start thinking about or coming up with solutions for those questions. Thank you for your attention.